Hello, good morning, and well, I'm really sorry that I was not able to come in person due to some technical problems, and I'm really thankful to Felix and staff that, who made it available for me to participate online. So my name is Mikhail Shugay, and I'm from Genomics of Adaptive Immunity Lab from Moscow, Russia, and also from Central European Institute of Technology in Brno, Czech Republic. And the title of my talk is Towards a Comprehensive Software Framework for Annotation and Comparative Analysis of T-cell receptor repertoires. So most of time in the lab I spent analyzing T-cell repertoire sequencing data, and I'm mostly a pure bioinformatician. And basically, a while ago, I came to the following conclusion. A typical analysis that I was performing is mostly based on three different methods. You are either looking at some basic characteristics like variable joining, segment usage, spectrotyping, or like repertoire diversity. You can look for clonotype sharing, just compare the T-cell receptor sequences across repertoires to see which repertoires overlap and which are not overlapping well. And you can, of course, use repertoire sequencing for tracking individual clonotypes. This is very efficient in monitoring minimal residual disease, but you can also apply it to vaccination, etc. But in my humble opinion, all these methods, they utilize only a minor fraction of information that is really encoded in the data set. So basically each of the million of T-cell receptor sequences that we obtain, it encodes some antigen specificity, and it would be really nice to know at least to estimate the antigen specific profile of the repertoire. But the problem is the following. Well, for example, comparing yeah. Okay, is it fine now? Okay, fine. For example, comparing with the metagenomics data, if you look at what people do when they are annotating the metagenome, they have a typical sample of thousands of operational taxonomic units, and the extent of known sequences in GenBank is enormous. You have like several millions of annotated sequences, and each of these bacteria are well studied. But for T-cell receptor repertoire, you have like 1 million different clonotypes in your sample, and you have only 40,000 sequences in the GenBank, and most of them are, of course, not annotated. They are not connected with some antigens. So the work I've started a while ago was to build a database that will collect the knowledge of antigen specificity of T-cell receptors that is documented in the literature. So basically to store the data in the format that will allow to see which T-cell receptors recognize which peptide in which MHC context. And we have built a framework to aggregate this data, and we are now manually processing the literature. So we have a Git-based framework for accumulating the data, and we're also in contact with several laboratories to share their private data on annotated specificities of T-cell receptors. And we've also developed a standalone application to annotate the repertoire sequencing data and a web-based browser that is also up and running here. It has some REST API with it and can be used to either look for individual T-cell receptor sequences or to annotate the samples. And the current release of the database, it was done several days ago, and it contains 33 manually parsed papers because it's really hard to extract the information. You have to copy the tables, to look into the methods, to understand how the T-cell receptor specificity was confirmed, because next you will need to rank the sequences based on your confidence. For example, if you have a tetramer sorting and one of the T-cell receptor sequences for the same tetramer is found in 95% of the cells, and then another in 5% of the cells, it's likely that this 5% can be simple contamination. So these are the statistics right now, and you can see a number of different antigens, and the spectral type is rather like bell-shaped for TCR beta. So we have a rather uniform sampling from the literature. And when we try to apply this this method, we have done it first for our data sets. The aging data set, we had around 100 individuals of different ages, and we've just looked for the number of CMV EBV specific clonotypes and their size, and you can see that while there are some clonal expansions in people in old age, the diversity of the specific repertoire is falling, and also the convergence, the number of nucleotide sequencing coding for the same amino acid sequences. Those receptors with the same amino acid sequences, they have likely the same specificity, but this shows that you have some sort of stochastic selection, and it was there was like a theoretical paper on this that showed that the repertoire diversity decrease that is well known in old age is associated with stochastic selection of different specific T-cell receptors that are specific to the same antigen because of cross-reactivity. And next, if you look here, this was done with a rather old version of the database that had 400 CMV EBV specific clonotypes. And you can see that actually the number of matching clonotypes, exactly matching clonotypes, is really small. It's less than 100. And even if we will increase the database and have 4,000, the number will be likely 1,000 of them can match, but still it's 1,000 out of 1 million clonotypes. And this shows that exact matching may be not the best method to annotate the repertoires, and one need to understand how to solve the central problem of how to tell whether two T-cell receptors are likely targeting the same antigen. And it's a really hard task. For example, here you have 3 different antigens, and each of them, the first two have 2 pairs of alpha-beta receptors, and the other has 3 pairs. And if you look at the similarity of this, for example, TCR beta here and here, you can see that there is almost no matching at all. There are so many mismatches, and it's likely almost impossible to tell something based on sequence similarity. But if you will try and go to the structural data and look at the contact maps, the contacts identified from the T-cell receptor peptide MHC structures, you can actually see that while the sequences of CDR3 regions are different, the patterns in the contact maps of CDR3 and the antigen are almost the same. You can see these dominant amino acids of the peptide that have most number of contacts, and the scale is the distance from the structural data. So if you look at another pair of different T-cell receptors for the same antigen, you can also see that the patterns are almost the same. And here you can also see that the patterns are almost the same. So the idea was to go to structural data and to try to find some sort of statistics that will help us to tell about whether the specific TCI is specific to a given antigen, and maybe just some bulk statistics to annotate our repertoire. So we've developed another pipeline to mine these TCR-PMHC complexes, to annotate them, to mark the variable joining segments, CDR3s, and to align, to identify MHC levels, and also to perform some basic sort of computation, compute distances and compute point energies using GROMACS. And there are several lessons learned from the data. First, if you will look at the number of contacts, you can actually see that the number of CDR1 contacts in some cases is actually not that small, and it's comparable to number of CDR3 contacts. So you always need to account for the variable segment, and it is better to have a full-length TCR sequences to actually read the CDR1 region to better define the antigen-specific properties. And the other thing is a distribution of contacts across the TCR and antigen, distribution of contacting amino acids. You can see that for TCRs, all the contacting amino acids are mostly in the central region, and for the antigen, the contacting amino acids, you can see the polarity. The alpha chain binds first and then the beta chain in most cases, while there are some counterexamples, and you can see the same for CDR1. So basically, this is a range of amino acids that have the most contacts, antigen contacts associated with them. And if you will compare the number of contacts between alpha and beta chain, you can see that there is actually a negative correlation. So there are some complexes that have more alpha chain contacts, and there are some complexes that have more beta chain contacts. And actually, looking at the energies, you can really see that if you will compare complexes with the same antigen, and some of the complexes will have higher contact energy, higher total contact energy of alpha chain, and some will have higher contact energy of total beta chain, but the sum of those energies will be almost constant within the TCRs that recognize the same antigen, but this data is not shown here. And you can also see that there are several physical properties of amino acids that correlate with the frequency of contacts for a given amino acid. And some of those properties, the core property from protein-protein interaction interface, can actually distinguish the contacting amino acids of CDR3. And you can also simply build this matrix, the preference of the amino acids to contact with each other. These columns correspond to an antigen, and rows correspond to CDR3. And you can see that there are two clusters. This one is the amino acids that typically have high number of contacts with an antigen. And you can then try to check it on real data. And to check it on real data, we can go to this recently published study. It was done in Alexander Rudensky lab, and we performed some bioinformatics for it. It was sequencing of TCR alpha repertoire of mice. And there was an experiment with CNS3 knockout model. CNS3 is an enhancer that is controlling FOXP3 expression and T regulatory cell development. You can see that, well, when you just overlap repertoires based on the exact CDR3 matching, you get a very nice picture here, very nice separation of tissues and subsets. But I decided to check if you can use the properties, the physical properties of CDR3 and, for example, CDR3 length to have the same separation of subset. And if you will check, look at the fraction of those amino acids that I have shown you previously that have typically high number of contacts, you can see that you can divide the populations, the regulatory population, the naive population, and the effector population very nicely. So you can see that paradoxically here, the effector T cells, they have a lower fraction of these high contact amino acids. But this can be explained by looking at human data, because we are looking here at alpha chain. And when we look at human data and beta chain and compare the memory, the naive repertoire and memory repertoire, we can see that the fraction of high contact amino acids in TCR beta is actually higher in the memory compartment, which is quite logical. But as these are all of these 3 points, 3 blood draws from the same individual, and they are CD4, CD8 memory naive cells. So next we will go to alpha chain, and these are the same samples but sequenced separately for alpha chain, and we can see exactly the opposite, the same picture we have seen in mice TCR alpha chains. So basically memory have a lower fraction of high contact amino acids in TCR alpha. And when we plot the same samples against each other and look at the number of TCR alpha and TCR beta contacts, we see the negative correlation. So it's like the same picture that we have seen from the complex data. There are some TCR PMHC complexes that have more TCR alpha contacts, and the others have more TCR beta contacts. And basically, when in naive compartment, we have a bias towards TCR alpha contacts. In memory compartment, for this individual, at least, we have a bias for TCR beta contacts. Well, this basically tells us that we actually need to do, of course, paired chain TCR alpha beta sequencing to understand this better. And yeah, we can also apply some insights from our structural data to look at the epitope databases and the epitopes. And for example, this is data from HIV escape epitope database. So there are some escape mutations that abrogate HLA binding, some escape mutations that abrogate TCR recognition, and some mutations for which it is unknown why the immune response is diminished. And you can actually see that HLA mutations are distributed by the sites of the epitope, and TCR mutations are in the center of the epitope, like in a good agreement with this picture. And we can also try to see if there is some negative selection based on this contact matrix observed in TCR escape variants. So we can define something called a recognition profile of an antigen amino acid. So it's just this profile, how this amino acid is recognized by different amino acids of CDR3, and we can compare these profiles between 2 amino acids and tell whether they are similar or not, like by computing correlation, for example. And next, we can also subdivide this other set based on the positioning and tell that this peak is likely those that have lower TCR binding, and these 2 peaks are those that have lower HLA binding. And when you compare these different subsets based on the similarity of TCR recognition profiles, you can see that there is a real decrease between those that escape immune response by not binding to HLA and those that do not, as do not bind to TCR. So our hypothesis looks like to be correct with some statistical significance, and there is some negative selection. So when the mutation occurs, you better mutate to amino acids that it will not be recognized by the same CDR3 amino acid if you want to escape the response. And basically here are the conclusions. So when you create a database of TCR receptors, you can make it large enough, but you need to define the rules of TCR matching. And this is an ongoing work we are currently doing, and we also try to apply some machine learning. And on the other hand, you can use, you need structural data for this, because it looks like everything is encoded there. And raw matching of amino acids, like sequence alignment, is not that good, the conventional one, to define the TCR matching. And yeah, you can use structural data as is to annotate repertoires and epitope database. And there are 2 issues we can see from structural data. 2 issues from structural data that we likely need full lengths for TCR sequencing, because we would like to know the CDR1 antigen contacts. in future, and we of course need prior TCR alpha beta sequencing right now. So that's it. Just to thank my lab and our collaborators and our other collaborators and the VDJDB I'm now working on is an initiative from the APRIM project. It's our Horizon 2020 project. And that's it. Thank you very much for your attention. Hope you heard me okay. Everything was fine.
What I'm going to try to do is to go back and forth between the mic and the speaker on the computer and hold the questions and then mute ourselves and get an answer. So does anyone have questions for Mike? Probably have to turn on that mic. And then turn it off when you're done. Oh, okay.
So this is a very general question. As a BCR person looking at TCR research, I've always thought there was a greater focus on CDR3, and so it was interesting to see you emphasizing the importance of CDR1, and I've also felt that there was less interest in partitioning into VDJ, or at least I know D is difficult with TCRs, but identifying the V gene. So my question actually, since we've been discussing the adequacy of the germline repertoire for the BCR, is the TCR germline repertoire adequate at the moment? What do you think might need to be done there?
Hello. Yeah, do you hear me? Okay. Yeah, of course, with the B-cell receptor, you have hypermutations, and they occur frequently in CDR1, CDR2, and this is of great importance. And in T-cell receptor sequencing, there are two problems. First, we, of course, don't know all the alleles. So what ideally should be done, you should take a pool of blood from lots of donors and make a full-length sequencing, correct it for errors, and identify all possible TCR alleles, variable segment alleles at least. And right now, the data we have, it is mostly from short reads, so you can't even get the allele resolution. You get like 30 bases from variable segment, and you can get good resolution to certainly tell whether it's the same, it's one or different segments, and you can even tell that you don't know about alleles, and there is no software to identify alleles. For example, there is some nice software packages like Tiger for B cells, but for T cells, we have, I think we have not dealt with this question, and we are not focusing on it enough. So you are correct, but the time will show. The full-length data will show if we really need to focus on it or not, or it's very simple, or variable segments are very similar in T cells, and there are like not much allelic difference in CDR1, but right now I don't know really.
So I guess one comment, one question. So one is, I think Tigger could actually be used for T cells. I know some people have been using it on T cells, so that's something could be done. Question, unfortunately, I think Bjorn's not here this morning, but I would like to ask, we heard a lot yesterday about IEDB, which is another established database that captures T cell and B cell receptor sequences with known specificity. And I'm wondering if you can comment a little bit on how the VDJ-DB that you're describing, how that relates to, or how that's different from IEDB.
Hello? Yeah, the sound is on. Okay. maybe I can move like this and it will be better. Is it okay? Okay. I shouldn't be too close to my PC. Okay, so regarding the IEDB, well, I haven't checked it a lot. So basically, perhaps these two databases can be complementary, but I don't know if it really contains all the required information, because we wanted to compile not only the specificity, but also the MHC alleles and the peptide sequence, and most importantly the technical details, because we really need technical details to verify the reported specificities, and we wanted to make it easier for people to submit the data by using this the GitHub, and by like making this sort of infrastructure. But I currently I think I cannot answer this question completely. But I think when I checked the IEDB, I've seen that there are some like some sort of things that I wanted to change, or there's some sort of things missing, but yeah, I haven't done really a big comparison on it. So perhaps I need to go and check it. But right now we haven't done any comparison on it. And I believe it's like mostly B-cell based.
In the B-cell structures that I look at, there are quite commonly contacts from the framing region, particularly framing region 3. And I wondered whether you saw the same thing in your T-cell analysis, and whether we might be missing something by not looking at framing region contacts.
Oh, there is a high possibility, because hello? There is a high possibility of it, because we haven't looked on the framework regions right now, but we definitely need to look at it. And right now we are doing this in order to well, basically study the extent of the difference between the number of contacts of variable segment with the MHC complex, and the number of contacts between variable segment and the peptide itself. So yeah, we need also to check frameworks. Perhaps they are more restricted to MHC binding, but this is a good idea, and we are right now just analyzing this data, looking into it.
Mike again?
Yep. Thank you.
Great. I'm glad that that went off. So hopefully we won't have as much echo for Chris Volmers' talk. So back in 2013, I just recently heard about repertoire sequencing, and boom, out came this really fascinating paper from Chris Volmers concerning measuring the B-cell memory response using B-cell sequencing. So he wrote that paper when he was a postdoc in Steve Quake's lab at Stanford, and he's now moved on to a faculty position at UCSC, where he continues to do fascinating methods development and immunology research.
Fascinating. I like that. First, thank you very much for inviting me to talk here. Like, I just started my lab, and I'm always very happy about getting the chance to like shamelessly self-promote my research. I should also probably thank the American government for giving me a green card and allowing me to be here today. It should make my job a little easier. Yay, immigration. So I should, I'm giving all my slides away. I'll probably go. Oh, okay, that's what it is. So I call this talk Sequencing Full-Length Immune Repertoires, and I will explain later on what I understand under full-length. That's somewhat of a loaded term, I suppose. Let me start from where I was coming from. So when I was a postdoc in Stephen Quake's lab, I joined the lab and I talked a lot with a grad student at the time, Josh Weinstein, who published, I think, the first paper on immune repertoires in general, on zebrafish in particular. Incredibly smart grad student, and we discussed quite in depth what you would want out of an immune repertoire sequencing data set. At the time, he and basically everybody in the field was using 454, and that was even before they really expanded on the read length, and they never really overcame their sequencing error problem. You have lots of indels and just issues with the data in general. Chimeras like crazy because of the funky emulsion PCR you do during the library prep. So my marching order as a postdoc was to come up with an Illumina method to sequence immune repertoires. And again, we talked a lot. We thought about what issues could come up when you actually use the Illumina sequencing to look at immune repertoires. And you run into a whole type of new problem now using Illumina data. When you use 454, yeah, you introduce a lot of errors and indels, but ultimately it's fairly likely that if you have one molecule you start out with, you probably get one sequencing read or less for that molecule. Now using Illumina, it's super easy to just completely overshoot the number of molecules you put in with the sequencing read you get out. So you get 10 sequencing reads let's say you get 1 million sequencing reads, but you only put 100,000 molecules in. Now, simply as a function of the sequencing error, instead of 100,000 molecules, you now look at 250,000 unique sequences. And you can do the math, but ultimately every fourth or so molecule you sequence is going to have an error in it. And at that point, I thought, do I really want to deal with this? And for me, the answer was clearly no. I don't want to spend like a little time making the library and then spend half a year getting a headache over whether this is an actual somatic hypermutation or a sequencing error. And I probably will never know. So the method we came up with in the lab is fairly straightforward. So at that point, a paper had just published out of the Vogelstein lab. They used this for cancer amplicons, where they simply introduced random nucleotides, and this is now fairly commonly used in the field. I think I'll just go over it. So you introduce random nucleotides during the RT and during second strand synthesis. You purify those primers away, and then you can simply run PCR on the outside of those random nucleotides, and every molecule will be marked with its own unique sequence of these nucleotides, and then you can use error correction to get one error-corrected molecule sequence out of there. And that worked fairly well. There were a bunch of downsides, though. The biggest one was that while the variable region is fairly long, in order to use Illumina sequencing for this, we had to ultimately just plunge a primer or like a set of primers somewhere into the V segment. That ended up being the framing region 3. So while you did get a perfectly error-corrected CDR3, you lost most of the variable sequence. So while you could totally do VDJ assignment on this and then upload your VDJ assignment into a derivate AIRR compliant thrift-based database, to be perfectly honest, the V segment assignments, and especially the allele assignments you did on the data, was basically worthless. You might get the family right and maybe even the segment, but you're basically guessing on the alleles. But like just putting a V segment and allele call in there is basically, gives you probably a false sense of security. Regardless, what this method was really good for was tracking clones. So what we published ultimately on this in 2013 was a paper where we looked at people that got the flu vaccine 2 years in a row, and what you can see here, we tracked their clones. So 7 days after the first vaccinations to 7 days after the second vaccination. Ultimately, the whole paper was just a reason to publish these plots because I spent a completely unreasonable amount of time coding those from scratch. Let me know if you want the code. I'm really proud about it. So that happened, but the moment this came out, we were basically unhappy with the protocol, and we struggled for a while. And that was at a time where Illumina actually was, still releasing better sequencers instead of worse sequencers every year. I'm looking at you, Mini-Seq. Anyway, so we were hoping for the MySeq to ultimately get there. So the 2x250 was released and the 2x300 ultimately, and we tried to fit an amplicon in there somehow. So what we really wanted was an approach that would take an antibody heavy chain and sequence it from the beginning of the leader to far enough into the constant region to call isotype subtypes, and then error correct that. And if we thought 2x300 might be able to do this, and we ran a couple of experiments on this. And what ultimately happened was that the error rates basically got so high towards the end of the reads that we couldn't, at least reproducibly, get those sequences out in a way we were happy with. Error corrected, no focus on the shorter CDR3s. Like we wanted to capture really all CDR3s, all heavy chain transcripts, and error correct them at the same time. So yeah, that was our goal. Full variable regions, error corrected, enough of the constant region to call isotype subtypes. Because we had multiple studies. We had one study where we looked at twins and V segments, and then we had to kind of put the primer really close to the end of to the beginning of the constant region, so you lost the subtype information. Or we were really interested in allergy, so we moved the primer further into the constant region, but we sacrificed part of the variable region. So I wanted one protocol that can do it all, that basically gave you perfect, like even repertoire sequencing out. And the way I wouldn't set this up if I didn't think we had solved this. So what I was working at the time, so I always was interested in immune repertoires, but at the same time also about single cell RNA-seq. That was always like the second thing I was working on. And Illumina released this really cool reagent, well, they patented and everything, which they called Nextera, which is a library prep method where you can take a low amount of double-stranded DNA, and what that TN5 enzyme, which they branded Nextera, does, it cuts your double-stranded DNA and attaches an adapter at the same time. So when initially all these high-throughput sequencing libraries were done by shearing and repair, A-tailing, and ligation, now you basically get a 5-minute reaction using this TN5 enzyme. Now, the problem with this was that, the way Nextera sells it basically is incredibly expensive. So having a new lab, we were like, hey, let's make this ourselves. And there's the Sandberg Institute at the Sandberg lab at the Karolinska Institute published a beautiful protocol for making your own enzyme. So we made our own enzyme, and now we had $500,000 worth of enzyme in our freezer that worked perfectly, but we didn't really know what to do with all of it. So instead of applying this to single-cell sequencing, we figured, can we do anything on the immune repertoire front with this? So what we came up with here, and this looks maybe a little daunting, but ultimately what we start with is we take our RNA, we put our primers far enough in the constant region so we can tell apart all the isotype subtypes, and then at the very beginning of the leader exon. You can also do this with race. We have tried it; it works beautifully. You do your normal UID library and end up with a PCR amplicon that's too large to sequence on a MySeq. So what we do then now is we take our library here and split it up into 3 parts, 3 aliquots. One we leave alone, and 2 of them we cut with our TN5 enzyme, which we load with adapters that are like with only 1 adapter. So this is where this whole thing becomes a little funky. When we make our library, right, you have 1 adapter that's for your forward read and 1 that's for your reverse read on opposite ends of your molecule. Now, for one of these TN5 tegmentation reactions, we only give the TN5 enzyme the adapter for the reverse read. So whenever this cuts a molecule, what you end up with is 1 part of the cut molecule that won't sequence on Illumina and the other one that will. So that's compatible. That contains 1 UID and both adapters you need for sequencing. On the other end, we do the opposite, where we load the TN5 with the other adapter, and we end up with the other end of the molecule usable. The cool part about this is that simply by adapter design, we ended up with 3 perfectly sequenceable libraries without doing any kind of enrichment. So you can do the TN5 reaction, you PCR it up, and it's ready to sequence. So no like funky biotin pull downs or circularizations, which other protocols that do like over-length sequencing these days use. And this is like a 1-tube reaction. It's really neat. But ultimately, what you end up with is 3 types of data. This might again be a little funky, but you end up with 3 types of read pairs. The first read pair is coming from the amplicon we leave alone, where you have 1 read for each end of the amplicon that defines our ends. The other libraries are anchored on one end, and then the other read is kind of floating around somewhere in the amplicon. But it's still anchored on one end. The other library is anchored on the other end, and the other read is floating around. If you quantify this, it's going to look somewhat like this, where you see the read coverage of your amplicon. This is your anchored reads. These are your reads that float around. So they are spread somehow throughout the amplicon. If you looked at just how this would look looking at text editor, this is one example of one molecule. Now the trick here is that both UIDs that you have at each end of the molecule have to be sufficient by themselves to mark the molecule. So they have to be about 16 base pairs long or 12, something where you can for sure say this is molecule A, looking at either of the molecules on either end, because your cut reads will only get one of the 2 of them. So what we really take advantage of is this funky difference for Illumina sequencers between the clusters it can make. So it can easily make a cluster of a 600, 700 base pair amplicon. It simply cannot sequence it all the way through. So by sequencing the uncut molecules and those 2 libraries of cut molecules, we can then go about and assemble every molecule into a perfect sequence. We do this, and a grad student in my lab, Charles Cole, wrote this. It's an assembly algorithm entirely for our little pipeline, and you might think that it's silly to name an assembler only you use, but if the name is that cool, you kind of have to name it that way. So the idea was we had these groups of molecules. We knew all these reads come from one molecule. We wanted to assemble them, and the first impulse was fairly straightforward. We would just use an off-the-shelf assembler like Spades or Velvet that have settings that might be able to do this, but it turned out they were super slow, and they made mistakes, clear mistakes that you can tell when you look, hey, this should look like a V segment, but it really doesn't. So we took advantage of the knowledge we had about each individual molecule. What we knew was, we knew the ends simply from our complete amplicons. So we knew where our amplicons ended. We knew that each read contained 1 anchored read and 1 floating read. So ultimately how the algorithm works is, and we knew that you only should get 1 contig out because this is 1 molecule. So we use our anchored reads, and then we k-merize the floating reads, and we just extend one end until we hit the other end. It's a very fast assembler. There's a few 100,000 reads in an hour or so. So it's cranking away. It's written entirely in Python. You can get this from GitHub or from our lab webpage, which includes basically all the other. Basically, what is on this web page is a pipeline of scripts that will get you from your raw data to the processed FASTQ. We thought, since this is somewhat of a funky molecular biology setup and the data structures are going to be weird, I want people to use this, so we basically released all the scripts that will take you from your raw data to your processed FASTQ that you can then throw at IGPlast and deal with that yourself. This also includes a couple of tweaks we did. Since we now deal with 32 random nucleotides instead of 16, we kind of thought we had to make some allowances for errors in the barcode. So this allows for some mismatch between the barcodes and does this in a fast way, because as it turns out, comparing, 10 million IDs to 10 million IDs takes quite a bit of time. So there's some, even with a fast computer, so there's some allowances for this. This should fairly comfortably run on any kind of desktop computer now. Okay, data. So again, up there is what I think, a full-length IGH molecule should be. It's about, the longest it should get is around 580 base pairs. So you see this length distribution of molecules we assembled. It doesn't seem, this is not running out of steam. The thing can assemble longer stuff. It's really not limited to IGH. If you want to look at 16S, for example, this is probably your thing to use. On the right, you see the kind of coverage you need to assemble one molecule in raw reads. So the coverage that's really decisive for your assembly success is the coverage you get out of your TN5 libraries, your A library and your B library. And here, once you have about 10 reads of either, you're getting into the blue. So this is where you start actually assembling efficiently. And this is 2 by 150 reads. So this is done on a HiSeq 3000. This is not used, and which makes this thing quite cheap, to be honest. Like even if you sequence 2 by 300 on a MySeq, the one thing I haven't mentioned so far, 2 by 300 runs are quite expensive. And the thing is, nobody does those unless you are, like looking at this kind of stuff. You probably have to always do a MySeq run by yourself. The nice thing about doing 2 by 150 HiSeq runs, you can probably jump on somebody else's run or do other stuff on a 2 by 150 run, where a 2 by 150 run is probably the adequate run to use. So if you get about 20 reads plus a few of your, so 20 to 25 raw reads give you one molecule, an error-corrected full-length molecule. If you do the math on the price of the sequencing run, this is still cheaper than doing a regular error-corrected MySeq run by probably a factor of 2, depending on how exactly you hit that coverage. This is a library that you can see we massively over-sequenced on purpose to see the characteristics of the library. Now, since we're assembling something, you could think there might be assembly artifacts or overall errors that were carried in there. So what we did to figure out whether that was the case is we used a control library. That was a 2 by 300 MySeq run on a shorter amplicon. This is our TMI-seq approach. And we looked just at the CDR3 lengths. We figured if we have any kind of assembly error in your CDR3 length, you wouldn't see those nice steps of 3. You would start seeing noise. And that wasn't the case. We also looked at V segments. I didn't put that figure up, but there doesn't seem to be indels, at least not at a rate that's in any way higher than a normal error-corrected MySeq run. Now, why, I mentioned initially, why do I want to do this? I didn't do that. My buttons don't do anything. Okay. No. I was about to show data. I mean, this is what should have happened when I clicked the full screen button earlier on. I'm clicking all the buttons. Ah, here we go. I got it. Now I actually have to show data. There's not a lot of data in there, to be honest. So the first thing we wanted to do was isotype subtypes, right? So we looked at 3 people. We sequenced about 100,000 heavy chain molecules from those guys, and simply like, can we reliably look at these isotype subtypes? We just split them up, and you get a fairly reliable or fairly reproducible amount of isotype subtypes you can tell apart. Again, for this you have to go about 100 or so base pairs into the constant region. So one cool part about the data, you get isotype subtypes. The next one was, and I honestly, I made this presentation before I came here, and now I feel this is like a hilariously crude way of analyzing, like these segments after looking at Figure and IG Discover and all those kinds of tools, and I will use those once I, have the time, and it's going to happen. When I say it's really crude, is my idea was, hey, we can perfectly and accurately, unambiguously assign these segments now. What would interest me there? I actually lately, actually got interested into the locus itself, and there is a couple of parts of that locus that are known to be, a variable between people. And due to your work, Corey Watson's work, who actually sequenced the locus, in like a heroic effort. Now we actually know that there's quite a bit of variation. Can we actually look at different people, just at the repertoire, and see whether they have those deletions or not? So I simply, and again, this is why it's screwed, I basically gave them a colored box if they had an allele and the white box if they didn't. And this was like a perfect match. So every time a box is gone, that means they either have an allele IMGT doesn't have in their database, or they simply don't have the V segment. So that's just as a disclaimer up front. So the easiest one to look at is probably V7-4-1, which I think everybody that has looked at immune repertoire data has seen that thing missing in people. And it's the same thing here, like individual 1, I should say, each row is an individual, each column is an allele for that segment. So individual 1 has an allele, individual 2 has an allele, but individual 3 simply doesn't have it. It doesn't show up. And that is like night and day. So individual 1 and 2 have like 2,000 out of 100,000 of this guy, and this dude has literally 0, 1, or 2. It's like the noise level is really low. And I went just through these different, like structural variants to look at, and this guy, these two segments I can't see, and that might be because they're identical to other 1-69 alleles. I honestly don't know. But this dude, V2-70D, we actually detect in 2 people, but not in the 3rd. And that story repeats itself over and over. Like here, both V segment and this structural variant, we actually pick up in one individual and not the other. So if you go just variant by variant, you can actually get an idea of the haplotype of these people. And again, we got incredibly lucky because we ran into people that actually seem to be homozygous for these events and not heterozygous, because then the whole thing would fall apart. This particular event, everybody seemed to have. This was really only interesting because once we looked at 3-30, where some people had more than 2 alleles, other people only had 2 alleles. So maybe if we sequence this deeper, or we actually sequence the locus, we might detect those duplication events. I'm here. Oh yeah, this is my favorite one. That's why I kept it for the second slide. So here there's two distinct haplotypes, one that contains 2 V segments, one that contains the 2 others. And this is the one where I actually believed the data. So we have two individuals that have the V segments of the top variant. Well, one individual had the 2 alleles on the bottom. So that's actually where I was like, okay, this probably is real. And then that one particular individual also had a different allele for the V segment right next to it. So then, again, this is a very quick analysis. And when I tell you there's some V segments that seem to have a funky pattern, again, this might be due to simply the allele that, the people that don't have this V segment showing up, not being in the database, or they're truly being simply a deletion that we don't know about yet. Because honestly, that locus is messed up. I don't think we know half of it yet. So just to summarize, once you look at all of these in the locus, you see that this is a mess. So I'm really looking forward to not looking only at 3, but maybe at 30 people and see what is actually going on, how many different haplotypes are actually around. Okay, summary. So what we developed is an approach that basically doesn't have to make the trade-off that other approaches have to make. It's a little bit more complex up front, as in you make 3 libraries for your sample instead of 1. But the actual additional wet lab work you have to make is maybe 3 hours per library. And that's highly multiplexable too. So the effort that you have to put in up front is not that high, to be honest. The approach is highly accurate. It basically is accurate as any error-corrected approach, to the point where at least the calculated quality score per base is so high we can't encode it in FASTQ anymore. You get what I call full length, and now I'm going to qualify this. I think a full length repertoire should give you the subtypes and the complete variable region. The moment you basically give up on that, you will not know the full information about the sequence you look at. I already said this is easy to implement. We supply the full informatics pipeline that we use, but I should also give you a disclaimer on this. This was written by us for us. So the moment you change the adapter, your computer is probably going to go on fire and say, I'm not dealing with this, because many of those things are like coded in there, and the documentation is like yeah, no, not really there. But it works as long as you do it exactly like we did. And if you have, it also, and this is more of a side effect than a real feature for you guys, it works on any amplicon. So you're not limited to immune, like you can do heavy chains, light chains, TCRs. We have done all of those and they work. Light chains are almost trivial because they actually, I mean, they're shorter, so they're much easier to assemble. It works with race. So if race is your protocol of choice, as long as you figure out how to attach molecular barcodes both into your TSO as well as into your RT primer, you're good. So we have done this and it works. Yeah, I think that's basically it. And I would probably take your question at this point.
Hi, great talk. I completely agree and support the quest for full-length information. I think it's really important. But why not PacBio? You get a single molecule, highly accurate, contiguous read with no assembly required.
I'm a big fan of long-read technology, right? We have a nanopore thing in the lab. I have worked with PacBio data, and I just say it's not highly accurate. Your error rate is still about 10%, and you could probably use molecular barcodes and oversequence, but then you have actually a lot of fun actually associating those molecular barcodes. So yes, you could use PacBio. There's a couple of issues with PacBio though. It has an incredible length bias, and I would be afraid that even in the realm of just tens of bases, that the short molecules would just be favored on the sequencer. So I would hope a protocol comes out, and I think if it does, it should probably, at this point, we have a hack here where we can actually compare secreted and membrane-bound antibodies by simply priming the RT of either of the exons. So we can differentiate those into different library reactions. I think if you go to PacBio, you should really probably start your read right there, like at the secreted and membrane-bound exon. And we totally tried that too. The problem there is that your secreted isoform, if you want to sequence them together, the secreted isoform is about 300 base pairs shorter than your membrane-bound isoform, and then you really only see secreted isoforms. Like that's how bad the length bias on PacBio is. Yes, sorry.
So I would strongly disagree with that statement, and I would say that there are protocols out there for both molecular prep and sequencing of short amplicons, short for PacBio, meaning anything under 3 kb. As well as a very highly accurate circular consensus sequencing algorithm to create the intermolecular consensus. And I'd be happy to talk to you offline about it because I've actually done the work with my own hands, so we should chat.
Oh, absolutely. And in no means do I want to trash talk PacBio. I'm actually, as I said, I'm a big fan of long-read technology. I'm even with the circular consensus, and now I'm saying this when you don't have a microphone anymore. But even the circular consensus, like right here, the error rate is somewhere between 1 in 100,000 to 1 in a million, right? Like it's to the point where maybe 1 in a few thousand reads has an error, and yeah, it's. I think ultimately all of this is going to switch to long-read technology, and I would have hoped, and this complete talk would be completely like. Irrelevant if Illumina in the last 3 years had ultimately published their 2x400 read length, then I would. This was very much born out of a necessity for us, so that's where that was coming from.
So we have a nice method that anyone can have if they want. We've been using for a few years now. Gives you the full length. It's race-based. Gives you the full length IG using the 2x300 kit. When the 2x300 kit was working fine, it was beautiful. We found a workaround using 2500 in rapid run mode. It works fine. It's really easy. PacBio, done some virus sequencing, HIV sequencing. PacBio looks lovely. And with Brandon and Melissa, we recently did some paired, linked heavy and light chains. And I would say the error rate is as good, if not better, than Illumina.
I don't see your question there at all.
If anyone wants our protocol, just come and ask. It was a comment, not a question.
I noticed that about halfway
Danny doesn't
through.
ask questions, no. So I'm a little curious. You said you're doing 580 with 2 by 150 times 2, basically, right? So I said your amplicon is up to 580 nucleotides, and you're doing it on a 2 by 150 with the one and at the other end.
Yes.
So I'm wondering, do you see any effects of sort of pushing up against that total length when you're trying to do the assembly, or you said the assembly, any assembly bias, I guess, for longer sequences versus shorter sequences? So you showed that the overall distribution looks the same, but I'm wondering if that heat map of assembly, like if that looks different for the longer reads, maybe.
If you're skimping with the coverage, yes. So if you have maybe, if you're not at this like 10 by 10, right, where you have 10 reads on each way, then yes, you'll see a shorter being assembled earlier, as in shorter reads.
The yellow in the bottom left is maybe mostly the shorter reads then.
So we looked at this, and I had the same graph actually looking at different read length, and yes, there's a coverage. The coverage requirement for shorter molecules is a little lower, but once once you sequence the library to a level that I would deem sufficient, that goes away. So I guess you have. Let me think. So we haven't identified them as an issue because at this point we kind of figured out how deep we have to sequence them. But yes, if you didn't sequence them deep enough, you will preferably assemble the shorter ones.
Do you, so you're scaling your read depth or your request from the MySeq to based on how much input material you're using?
Yes, so we did a bunch of experiments up front where we basically used different dilutions of RNA from PBMCs at base, and then we figured out how many IGH molecules we get out of there. And so we basically gathered that experience, and now we run off of that. So we don't do an upfront quantification of the molecules. We basically figured it out by trying it. Yeah.
So I was very interested in your haplotyping.
They're incredibly crude, and I'm kind of ashamed I showed it haplotyping. Yes.
Well, yeah. So we published a technique 4 years ago on how to generate phased haplotypes,
and it's fantastic.
Well, yeah,
Thank you.
but of course it only works for 30% of individuals because, well, for those who don't know, basically what we did is we identified individuals who were heterozygous at a J gene, and that's almost always J6. And so then you can, if you look for rearrangements that are using J6, you can determine the two sets of E genes. But it seems to me that your technique gives a new opportunity because there's so much more allotypic variation. So if you've got your constant region and you look at your variants there, you'll nail everyone.
So we definitely look for that J6 allele, and all these people are sadly homozygous for the second allele. We said definitely because we tried to redo this, right? Having this data, we haven't found any SNPs in the constant region either that we could use for this. So that's why I also said I'm really excited about expanding this to many more people, which now actually fits into our budget when we have this approach. So simply to track these people down, and that's actually a question I always wanted to ask you. Do you think people that only have, that are heterozygous for the 3 allele, the J6 3 allele, are they a subsample of the population? Do you expect actually to find all the haplotypes? Like, basically what I'm asking is the J6, the 3 allele linked to haplotypes upstream in the variable region. Maybe linkage wouldn't work on a distance that long, but that was I always wondered about that method.
Yeah, look, I've 1 to 2. I've got no idea.
Okay.
Yeah, it's important, but I can't comment.
So in the data that you showed, how long is actually the constant region that you're using for identification?
About 100 base pairs. Okay. And that is so the reason I picked that particular primer location was for, I don't know whether that was I'll answer it really quickly and then I'll wait for your second question. We put the primers there for two reasons. One was out there we could differentiate all of them by one or multiple SNPs, and the other one was that was actually a good location to put primers in the same position for each isotype. Because there are certain regions where you would like, oh no, we can tell the IGAs apart earlier, but then the IgGs you can't prime off of there yet, or there's just a horrible position to prime off of because it's all Gs. So the reason we picked that particular location was a compromise between, because I wanted all the isotypes to be the same length, and I wanted to prime that. Anyways, yeah.
Okay, so the main issue is how sure are you that you can reliably distinguish IgG and IgG4? Because usually those are really just a very limited set of positions, and as far as I remember, all of them are annotated as SNPs. And when we went through the thousands genome data, then actually we were not convinced that we could really use those as identification.
I entirely went off on the IMGT annotation, and so there's several IgG4 and IgG2 alleles, and I tried to be able to tell them all apart. If you're telling me now that this is totally not true, how far do you think you'd have to go out?
Well, I think if you really want to capture the variability, you already said it. So I think you clearly would like to capture at least the hinge region, because there is a substantial variability in human populations, especially for IgG3, in terms of hinge exon duplications or quadruplications. Otherwise, yeah, probably end of exon 1, but in terms of technology, then it really doesn't make a difference anymore, probably.
I guess, I think end of exon 1, that's another probably, what, 200, 300 base pairs on top of this?
Yeah, they're usually 270 to 300.
Yeah. That might just still work with this. So we tried this out for stuff up to like 800 base pair amplicon. That still works. It's just the real limitation here is simply the cluster generation capability of an Illumina sequencer, and that just goes way downhill after about 1,000 base pairs. Before that, it becomes less inefficient, but it doesn't disappear entirely. So you have to just be mindful of how you pool your complete versus your digested libraries to make this work. So I'm not necessarily married to the primer set. I just felt like this is very, very much a modifiable, like library prep.
It's time for a break. So the schedule says be back at 10:45. Maybe let's extend that to 20 minutes, so 10 till. That sound good? Thank you. Okay. So our draft recommended. This is the Tools and Resources Working Group. Our recommendations continue to evolve, and I apologize for putting the wrong link in here, but this is the correct link now. So I thought we try to condense down the various recommendations into things that people might actually have an opinion about. So we're not going to go over everything. We're just going to go over a few select items. So the first thing was sort of stimulated by some discussion yesterday concerning how Dr. Sairedi and perhaps others are developing standards for immune repertoire sequencing, and this seems like a really great thing for our community. And so how can we pull together to facilitate adoption and dissemination of such standards? So clearly it would be great to be able to cooperate with some sort of company or organization to arrange for an AIRR set of standards. Let's see. This is actually, we were revising this at the very last minute. Let's see if I reload. This looks like maybe somehow it got converted to PowerPoint. Maybe this is an older version that got converted to PowerPoint. Okay, but maybe let's start that discussion, and I'll see if I can, pull up the web link. So let's see. So can we sort of kick off the discussion describing how we might be able to get a company or organization to distribute these things? So Danny suggested that maybe we could use ATCC, but I think that they're typically used to distributing single cells rather than sort of mixtures of them, so on.
I think the point is that, so if we want to establish standards that people can use to validate their library prep protocols and their sequencing protocols, it's nice to have RNA and DNA standards. They can be plasmids, stuff like that, but it's really also nice to have cells that contain expressed IGs or expressed TCRs, because certainly in our hands, probably the most important factor in the whole process is the quality of the RNA that you extract right at the beginning. We all have jobs, and we don't, well, I don't know, most of us do, maybe some of you are unemployed. We don't want to become the repository for cells that people order from us. I certainly don't want to do that. And there are repositories of cells such as ATCC, such as the NIH AIDS reagent program that already exist, and they have a great infrastructure. So what we thought we could do as a community is to test available cell lines, to make available cell lines, to make cell lines and deposit them in these repositories. And then as the AIRR community, we would publish our recommendations for the best set of RNAs, DNAs, and cell lines that you can use to test your libraries. So it's an AIRR approved set of nucleic acid or cell standards.
We also thought that perhaps we could motivate the creation of such a standard by making a reference publication about these are our core set of standards that we are encouraging.
Sorry. Has anyone had any experience with NIST, and would there be any opportunity that we could potentially work with NIST on this, because that's what the ERCC RNA sequencing community was able to do? Would it be a good idea to try to get in contact with NIST?
I mean, if that's what we need to, so the Thermo sells the mixes of standards for the RNA you said yesterday. So if that's what it takes for us to get Thermo to sell what we want people to use, I think, then yes, that's definitely something we should pursue. But I obviously know nothing about how to go about that.
So I have experience with NIST. I worked with them when I was at PacBio, disclosure for everyone based on our last conversation, developing a mixed microbial community that they're using as a standard for bacterial assembly and screening of bacterial things. So involving NIST would add a level of bureaucracy to the process, but they are very, very thorough and very good, and I know people we could get in contact with to start that discussion, so I'm happy to be an envoy in that way.
We worked with NIST to deploy the ERCC in our lab, and we found to be extremely nice collaborators and very willing to help. So I think they're good people.
Great, okay. Another thing I should have mentioned, and I'm not sure I communicated it clearly, as at least a starting point, the spike-ins of human that I set up, the mouse spike-ins are already available if you're interested. The human spike-ins, which were over the 85 different clones and several different germlines, or most of the germlines that are well known, that was done in collaboration with Novartis. I have made it clear, or signed the agreement with Novartis, that I'm free to distribute that spike-in set to anybody for non-commercial use. So you can request that from me, and I don't even need Novartis' approval. So once we've finished validating that, I'm happy to distribute that either as plasmids or cDNA. So yeah. Is anyone aware if NIST does cell lines?
The microbial stuff was mixed and then DNA extracted. They were going to provide DNA because it's more stable, less malleable. But obviously it's worth a discussion. They've worked on so many different things. I don't know the full catalog.
Well, if they do or don't do cell lines, the NIH AIDS reagent repository is an amazing resource, and it has everything from viruses through to cell lines. And I know the people who run it, and I can ask them if they are either interested in helping out with cell lines, if we find that NIST are not interested.
I think, if I understand, part of the issue is that we're not talking about just cell lines, but like actually mixes of cell lines probably in defined ratios, and so.
Okay. I mean, you could do that, but I think it's easier. I think it's easier just to have individual cell lines, and then people can count the cells and mix them as they like.
I don't know how far out this idea is, but we've been using as a standard in our RNA sequencing pipelines actually just the same donor again, so just using frozen PBMCs from a donor. We got a pheresis from with 100 billion cells and just running like 100,000 of those in every run. So, I mean, the kinds of controls, if you're mixing cell lines together, you're trying to mimic the complexity. So maybe there's an argument for actually just have frozen PBMCs, which would be T cells or B cells, and allow to potentially control for all kinds of, like, I don't know, germline biases or so in your approach. Obviously, it's a different topic, right? So with the very well controlled one, you really have a very nice positive control, but if you actually have a donor frozen down cells, which also, I mean, okay, it's somewhat simpler. Frozen PBMCs, distributing that's easier than maintaining cell lines.
I think that's fine on an individual lab basis, or maybe a few labs, and we do exactly what you do for that reason. But as a resource for the whole community for years, you could theoretically keep bleeding the person, but it'll run out at some point. I like the idea of using PBMC. I like the idea of cell lines because you can take your cell lines and spike them into the PBMC and get a lot of information that way. So it doesn't have to necessarily be the same PBMC donor.
I would also add one of the things we talked about within the working group last night is the idea that we might need different sets of benchmarks for different purposes. So, and this may be another reason to do individual cell lines that people can mix in by themselves. But just, we have a lot of different use cases. People are looking at sort of general property of repertoires or its specific lineages and many things in between. And so it may be that one benchmark doesn't fit all. And so, some of these ideas like PBMC may be very good for some applications and less useful for others.
There was a suggestion which started off as a joke, but it's not so crazy, but there was the idea that we could make these available through Amazon. And I think somebody knows someone at Amazon. Who was it? He was sitting next to me.
I think, would it be Prime eligible?
Well, so I want to claim this now. So for primers, then, yeah, Amazon Primer. You never know. It might be in the future.
I was just going to ask, there's a lot of immortalized cell lines for B and T cells. I was wondering where you think those might fall. They have different expression levels. They're all different, but they are renewable resources. So I was curious what the thoughts were there.
I think as long as they're generally available, I mean, so of course you want to keep the strain or the burden on the labs low if it's their job to get the individual cell lines. So meaning that everything should be S1, not S2. Having different cell lines with different expression levels is probably a good idea because it makes it easier to validate. The one thing is we should make sure that people really order the cell lines not from the repository. So ATCC or the other national counterparts, because otherwise they're using stuff they're thinking is cell line X, but in the end it's
unless they are cell lines that are AIRR approved cell lines which we validated.
Yes, but then we would have to sell them again. So the point is about having the repositories or the biological repositories take care of this is that we can just say, okay, this is the ordering number, use that. Mix it 1 and 1. We know that this is a memory cell. It only expresses tenfold less than this is a plasmacytoma, which produces much more.
We have a very brief discussion about species and loci. Or should we move on? Should we have a very brief discussion about species and loci? Which species and loci should have the highest priority? Done. Okay.
I'd love to see platypus.
Hey, don't talk about your private life here.
Couldn't we do a possum first? No. Okay. So, does anyone object if first priority would be human, mouse, BCR, TCR, and so TCR, alpha, beta? No. Okay. Okay, but you're probably also the only one who could actually create that resource.
We have that resource created already for Rhesus. Will be available in a couple of days' time. Session numbers released. So, but we are working on other species as well. So, in the near future, through techniques like ours, there will be an exponential increase in the number of germlines which are available. I don't think we should start putting limits on species now.
It was definitely not a question of limits. It was a question of priorities. So, just which comes first, that's all.
Just ask you one question. Have you given thought to the international nature of your community and whether you're going to mirror those resources in Europe, East Asia, and the United States, or are you just focusing on US repositories to start with?
So I can only speak for the German-speaking countries, but there the central repository basically has or shares the accession numbers with ATCC. They also co-validate basically the identity of the subline, so I think that shouldn't be a problem. And as far as I understood, for most EU countries, that's the same.
Okay, I'm Qing Zhang from Genentech, and regarding the NIST question, I know they have this genome in a bottle consortium that's looking at the, these reference samples and all the methodology and standards. So I would encourage AIRR to become a member of the Genome in a Bottle Consortium and collaborate with them to generate these standards specific for the immune repertoires. Another comment is I actually don't know where it would fit in these working groups. I felt there's a disconnection, general discussion between the exome or genome sequencing world, with this precision medicine, collecting a lot of individual samples and doing these things. They seem to just ignore the immune repertoire because it's, understandably not fitting into all the whole exome, whole genome computational analysis. And I think it's a huge waste of opportunity because there's nothing more personalized than the immune repertoire, and more important at this era of, cancer immunology, what Steve Rosenberg was presenting yesterday, how important AIRR should be part of those precision medicine efforts, and I don't really see a crosstalk between the two, and I wonder if this community as a whole can push that to happen, to not waste those precious samples collected and to have the data collected.
So we are talking about trying to improve our reference databases and germlines, and we'll get to that in a couple of slides. It looks like. So I don't think that fully addresses the question, but having fully mapped references or a really good germline database, I think, would help with that at least. But that's about the limit of to which I can speak to that question.
I'll go ahead and speak for Corey and myself. We've been working with Genome in a Bottle, PacBio before did work with Genome in a Bottle, and we have access to that data to try to use the whole genome sequencing that has been done high quality on 8 different individuals of varying ethnic backgrounds to try to create those references, and Corey actually is leading that effort with a collaboration with myself at Sinai. So I think there's a lot of data that's already out there that we can mine, and the people who have looked at it so far have not had the focus that we have and the tools that we have. And then again, I think, as she said, linking up with the Genome in the Bottle folks, who again, I know and could facilitate introduction, would be excellent as they choose the next samples and where they focus. I think that's a great idea.
So a lot of those efforts, 1,000 Genomes projects included, they are based off cell lines typically, because those are able to be immortalized. So if it is something that we want to do as a community to sort of link up with those projects, I think we need to have conversations with them about what kinds of samples we would want collected, because it's not standard to just get blood, for example. And as it's already been brought up, you can't just continually bleed people. And so, yeah, I would say that's where the disconnect probably lies. So it's great for sort of doing genetics and reference, but I'm not sure it's so great for, unfortunately, for doing repertoire stuff.
Maybe let me make a suggestion around, since we're talking about standards. Maybe we don't need to bleed people all the time. We have transgenic animals that we might be able to use for this purpose, containing all the same genes we're talking about. So.
Yes, I mean we can do this for mice, but we don't have transgenic humans. So. Okay, but then what's the advantage using that one versus a cell line? Okay, so the question is why not use transgenic mice which contain human receptors? I think the question would be whether there's any utility for using the live organ instead of having a cell line encoding the same receptor.
I would just say the variability. So a cell line is generally a single, whatever your transgenic, single cell line, right? So it more mimics the human situation.
Yes.
So just for clarification, what do you mean when you say cell line?
When I'm talking about a cell line, it's like a plasmacytoma. It's grown in culture.
So how variable, if you take one of those to your lab and someone else takes one to their lab, are you going to get the same results always? Okay.
So of course, I mean, with all cell lines, I mean, this is the thing why I said it's important that people get those cell lines at one point from the repositories, because some of these cell lines are older than half of the audience here in the room. So and have been propagated over and over, and of course also evolved. So we cannot necessarily exclude that that the receptor sequences are identical. But same thing would apply for a lot of things. So therefore, once we get them from a tested resource, it's rather unlikely that this would be an issue. If then suddenly 1/3 of the community comes up, oh, we found a new SNP or a hypermutation, okay, then we would find it easy.
It might help in this discussion just to clarify what we want standards for. I think there's an underlying assumption that it's for accuracy of sequences, which is true, but it's been brought up a couple times now that a standard for diversity might also be useful, and that would be different than for sequence accuracy or something like that. So we might want to just clarify the rubrics of what the standards are addressing in the discussion.
Yeah, absolutely. And let's see, I think that might be a good segue into the next slide, Eric.
And just one thing, the idea to have a mouse with a human sequence. There is a Regeneron that they have a mouse where they put the complete human immunoglobulin locus. So I don't know whether it will be.
That mouse is millions of dollars to license, so I think you can kind of exclude that.
I doubt, but not having the mouse, but I don't know, something that would be useful probably, whether possible or not to get.
So to try and move things forward a little bit. So yeah, I think we do need to define exactly what types of standards we use. Like I said before, they're probably, we need a suite of different benchmarks for different scenarios. And so one of the things we're talking about here on this slide is what types of data sets we want. And here we're talking more about data sets rather than wet lab standards, cells or RNA or DNA. But the same sort of thought processes apply. You know, what types of analyses are you trying to do, and what do you need to be benchmarking? And so for this step, you know, we think that about 2 or 3 categories. So you know, simulated data sets with different generative models, so that as opposed to somebody building a tool and building a simulation to test the tool and ultimately on the same generative model, and then having a few of these that are widely used will provide equal footing comparison. One of the things we noticed, so Simon Frost has established a list of evaluation data sets at the link shown here. And it's really very, very useful, but most of them are actually fairly small data sets, or with the exception probably of S22. They're fairly small data sets, and the simulated ones are fairly simple clonal structures. And so one of the things we think we'll need is to generate more complex simulated sets that can be used. And then we want to be able to test pipelines on real data, not just on simulated data, for the same reasons. And so we're thinking that we probably need to identify sequence data sets that are publicly available and where we can sort of trust the experimental process, so we know, that the sequencing was done in a way that we feel comfortable is a good way to sequence a library, and so on and so forth, and then to maybe identify a couple of those and encourage, as part of our recommendations, encourage everybody to test their new software against the same set of same sets of data. And one of the things the only one that we've identified so far is the S22 from Simon's list, and that's a really nice data set because it comes from a fully genotyped individual. So they sequenced the gDNA and identified all of the alleles that this person carries, or at least that's the claim. Sorry, I'm turning to. So the S22 is from a Stanford data set where they sequenced the genome of the individual in question and identified, at least say they identified all of the germline alleles that that person carries. And so there's a list of those that goes with the data set. But if people know of other useful standards data sets or data sets that might be interesting to encourage people to use as test cases, we would really like to hear about those, because that's we need bigger and more complex data sets to be able to really test some of these tools.
I should perhaps clarify S22. So that's from our 2010 study, where we identified, we inferred a deletion polymorphism involving 6 D genes, and then with shotgun sequencing we confirmed that the individual was homozygous for that deletion polymorphism. The genotype itself is an inferred genotype, but I'm very pleased to see that people are interested in using. I mean, I do think it's a useful tool, but the genotype is inferred from the VDJ rearrangements.
Okay, so this part of the problem here is we've got Simon. I'm referring to something that Simon Frost compiled, referring to so. A little bit of a game of telephone, so it's good to have that clarified.
Sorry. So coming back to the diversity, I think beside the annotation and evaluating tools, we also need to evaluate the quality of our data sets. So you can have, depending on the methods for the library preparation and the sequencing, you can have different results with the same computational tool for annotation. So do you have any suggestion on this? What could we do?
if we are willing to do that. We have this experience actually. We compared different library preparation and we got like different distribution of P genes or J genes or even CDR3 overlap, which is quite important. So if you do 3 replicates and you get only 25% or 10% overlap, is it that good? Is that bad? So what would be the threshold on this, and if we have some ideas?
So I don't have an immediate good answer to that. For this type of thing, right now we're only thinking in terms of single data sets. So sample resample is a really big question that, and that does go back to, like you said, to the library prep that we talked about previously. And yeah, I haven't done a lot of sample resample myself. I have done a little bit, and I know, just from a mathematical, philosophical perspective, it's obviously an important question, but I don't have a good answer for it.
Can I think this points to the need to establish somewhere where we can write down good practice, because there are things that occur to me like monitoring cell counts, how many cells you actually have in your sample and that kind of thing, which might help get consistency. But if we all, if we start putting some areas together where we can outline what we think best practice is and discuss it, I'm sure we can do a lot better collectively than we're doing individually.
So we have this data set available. They are not that big, but maybe we can share and see what we can do with this already on TCR. I'm a TCR person.
So I just wanted to ask a couple of questions of the group. And one is, for instance, with the minimal standards group, they had things that they were recommending solidly, like standards. And then there were other things that were recommendations that were a bit softer that had to do with kind of the standard of practice, right? But not necessarily things to so I'm wondering, do you have the same kind of, for instance, I'm just wondering, that would you say that if someone is going to develop and publish a tool, that it should be validated somehow? And if so, what that means, as opposed to saying, well, it'd be nice if you did this, and it'd be nice if... So that's one question I have. Another thing that was brought up, if I can speak out of turn or whatever, that was brought up yesterday by Simon was maybe your group would be a great one to suggest either a competition or a collaboration, I don't know how you want to put it, to get a whole bunch of folks together to, for instance, look at certain types of tools and compare them. And then finally, I think that your group is very well situated to do the soup to nuts description for the naive researcher about the biological, data gathering, setting up for the data gathering, doing the analyses, and also fitting into the larger sharing world. I think that one thing we haven't really discussed much is trying to put something together that looks like that, as opposed to just lists of standards and so forth. So those are just some comments.
So if I can try to do this and remember all three. So we do have some pretty clear and straightforward recommendations, especially in terms of access and sharing. And those are in our recommendations document that we didn't, based on some of the feedback that we've gotten so far, we didn't feel like needed to be spending time on today. Though certainly, what?
Maybe I'll just hop in. I mean, this was something that we described yesterday and Monday. So I mean, the requirement, the more required side is things like open source and like initiative approved licenses, being hosted on publicly available data, repositories with versioning, and so we went over that yesterday.
Okay, so the reason why I'm bringing this up is what exactly would people be voting on Thursday, right? Because there are a lot of more general things, and so that's why I'm bringing that out, to try to get the actionable things that we'd be voting on as well.
So I think that's what we've been talking. Those are the things that we've been talking about, for the last half hour or so here. So the second question that you had asked was sort of about the division between tools and resources, and I think that we have that from our internal discussions. I think that the tools side of things is pretty straightforward, and that's basically where all of our definitive recommendations so far are, where things have been very clear and we've been able to make succinct recommendations. And in terms of the resources side, we've had a lot of discussion about what resources are useful and how we can motivate people to develop them, because with maybe some exceptions, like Eric putting up the website where we have a lot of our discussions, like we're not going to be developing these ourselves. And so that's, seeing somebody like Sai who's already doing a certain amount of this work, that's what we want to get feedback on, and how can we encourage that and reward that, and then turn around and encourage the rest of the community to follow his lead, both in terms of developing resources like that, and then in terms of using these sorts of standards. Then what was the third question?
There was the challenge.
Oh, so we've talked about a little bit about having, John Sang yesterday talked about his Jamboree, which we've talked about doing something somewhat similar, although it's not planned or anything. It's an idea that we said, we might want to look into doing one of those, and it sounds very exciting, but we don't have any advanced discussion on that.
Well, so we've talked about a hackathon, haven't we?
Yeah,
so
that's what I mean.
Yeah, if there's a resource we'd like to have for the community, for example, for managing germlines, we could get together and hack. But I think perhaps, Jamie, the thing you were talking about that's also come up is the idea that we might have challenges. We could have annual challenges for tools to meet. So, for example, one of my preoccupations is I think the computation is getting a bit ahead of the science when we talk about clonal determination. I'm not even sure I know biologically what we mean by a clone, but there are lots of tools out there that will identify clonal families. So if we came up with a really smart experimental way to say, here's a sample and we know what the clonal families are in that, we could challenge the tool providers to see how close they could get.
And I think that's a key distinction too, because we talked about some of this last night when we were trying to figure out what standardized data sets would really be useful. And the truth is, we don't have hardly anything where we know the right answer. And even when we have simulated data sets, we're suggesting that we get people to use several different simulated data sets with different generative models, because any one simulated data set is not necessarily reflecting the underlying biological process correctly, because we don't. So having a competition right now is a little bit hard because we don't have a data set with right answers. But I agree with William that if we can find a way to experimentally generate that might be interesting.
Well, so I just want to bring up that one thing that was discussed last year was the idea of getting pair read data and then hiding the light chain, for instance. And then you could see whether or not you're getting your light chains out of your clonotypes. But anyway, it's just a thought. But I totally agree that I don't think it would have to be a competition per se. Like I said, it could be a collaboration.
Sure.
The point is, if we have annual meetings, perfect time to do that, and that's what we do in antibody engineering,
right,
for instance, with modelers.
Whether it's collaborative or competitive, you ultimately, like you said, you need something that we can sort of judge on. And it's also one of the reasons here on this last bullet point on this slide that we're talking about trying to identify publicly available data sets, BCR, TCR data sets, where we can say, you should really run your tool on this data sets A, B, and C, and we may not know the right answers because they're just regular biological data sets where maybe we think that they were well processed experimentally, but at least then, even without knowing the right answer, you can compare your answer to the one that I got and one that William got and one Danny got and one that Christian got. So.
Hi, my name is Sarah Taylor and I work for CloneTech. So we make kits and we have a TCR sequencing kit. And a lot of the questions we get are very basic. They're kind of like, our tech support team are dealing with questions like, how many reads do I need for my sample? And I think it's come up a few times that a lot of people in the community are quite naive to NGS and big data sets. And it has been mentioned that maybe some basic biological recommendations would really help. And that's certainly what we see, like really basic stuff that I think we're sort of skipping ahead of a little bit because we assume people have this knowledge. But I think there are big gaps in knowledge in the community.
Thanks.
That was a great discussion. I wondered if maybe we could just take a quick minute and talk about annotation format and maybe stimulate a little discussion. So personally, I help write tools for doing various annotation and clonal family inference tools. And every time VDJ-ML has come up, I've sort of said, okay, that's fine, because what I'm going to do is write something that outputs the change-o data standard and then convert it to VDJ-ML as needed using the yield groups tools. But that's, in a sense, that's a little bit silly, right? So essentially the question is, I mean, are there tool developers out there who intend to use features of the VDJ-ML schema that are not present in a tabular format, with change-o standard being an example?
Do enough people use these formats to know what is included in the one that's not in the other? Question from the audience was such as what?
Do we want to have someone from the VDJ-ML team?
Well, just an idle observation from the demonstrations yesterday. When we were looking at the query tool, I saw features in VDJ-ML like caveat numbering, caveat determination of CDRs. You know, there's a data format, but there's also what data is going to be present. You know, if I want to convert IMGT to VDJ-ML format, are you going to expect me to put caveat determinations in there, for example? That makes the conversion a lot harder. So, you know, we need to bear in mind schema as well as format.
Okay, so a few things that are extra in VDJ-ML that aren't in the tab delimited format is in the tab delimited format, if you want to do any nesting of information, you're restricted to putting it in a single column using commas or something, and that can get out of hand if you want to, you know, have, say, 20 annotations attached to one field. So that's one thing you can do with the XML that you can't do with the tab delimited effectively. There's a bunch of extra stuff in VDJ-ML concerning tracking how the analysis was done, what tools were used, provenance information that's not available in tab delimited format as well. And as you mentioned, the numbering. So again, when you do sort of a tab delimited format, you're going to have to restrict yourself to a specific numbering scheme or add additional columns, whereas with the VDJ-ML you can say, you know, this is the metadata that tells me what kind of information is contained in these other fields. So those are sort of the big differences that I see.
Are you going to move to it?
So in a way, we already did. So we made a lot of contributions to it, and our original standard was different than it is now, but now it's actually more based directly on VDJ-ML. And similar to the tab delimited output that they generate from that, it's not obvious because the names are different, but the actual information that we chose to keep was the same except for all the provenance info. So we are definitely going to add support. I don't think we're ever going to stop using a tab delimited file because sending an XML file to our collaborators is just not going to fly.
Just your specific question about cabot numbering. So VDJML allows for you to use any numbering system that you want and specify what that numbering system is, but it's not required that you populate any specific numbering system.
But my point was that it might become an expectation. You know, for example, if you give people, and I'm sorry to pick this, you know, this example up, but if you give people a query tool that lets you query on, you know, a CABAT field, a CABAT CDR field, what's in it, you know, then presumably it's only going to report that for those files for which it has that information. So, you know, as well as thinking about the format, we have to think about what information we expect people to provide in that format in different circumstances. That's really what I meant.
And just one other comment, not in response to what you just said, but I agree with Jason. People aren't going to stop using TSVs or CSVs. They're flexible and useful for many things. But the benefit to XML for certain use cases is that with the schema, it's self-defining. Whereas with the TSV, you can make changes. It's easy for, you know, it to become non-standard and not self-describing. So I think there will be uses for both.
Just an additional comment to follow up on what Lindsay said. I think our intention for VDJML is to be very flexible. You know, there have been quite a number of suggestions posted on the BTCR board, and we're looking into a number of those. Certain individuals have requested, you know, a mirrored JSON document type, and that's something that we're in the process of doing as well. So I think we aren't restrictive to the version 1, and you know, we certainly want additional input from the community at large, code contributions and whatnot. And I think it will help us fine-tune it and refine the document to something that will be very useful. So I don't think we're restrictive in terms of the content. And certainly that's one of the items that came up yesterday during my poster discussion. So certainly feel free to follow up with us and join in on the dialogue. And we'll be on the board as well, the BTCR board, and we'll, you know, be very receptive to hearing what you all feel in terms of the content and things you'd like to see.
I just encourage to embrace some of the things that have been recommended from the data group. I mean, there's a reason that that everyone started out using CSVs and then moved over to more structured definitions in their spreadsheet files, right? It's because you needed more information than you could get with a header row and tab or comma or space specified delimiters. The value that structure brings is that conversions become easy, but also embedding more information in there, right? So you can go with XML and it's bloated and it's really expensive to parse and it's really heavy, but you know what? It's very well defined. And there are times when, you know, if you're taking out a mortgage, you want a whole lot of structure around that. You want these things well defined. You want to know all the ins and outs. You want those written down, right? But I don't want to fill out mortgage applications to borrow $5 for lunch, right? Sometimes you need a mortgage, sometimes you just need a couple bucks. And the value of having these structured formats is that you can go back and forth and apply as much rigor or as little as you need. So just I would encourage not to throw out VDJML just because there's a lot of information in there, right? Use what you need, leave the rest behind.
So my lab kind of lives in the middle between like the wet lab and the dry lab. And we write our own tools and we do all kinds of stuff. And coming from my lab, if you design a tool that gives me a VDJML, an output that wouldn't be a flat file, I would categorically not use it. And that may sound strange, but I like to get output formats I can easily parse myself. And it's not like, you know, I came out of the genomics field. Those people are living happily with SAM files, VCF files, all flat file formats. They agreed on that, they use it, it's been for years, it's expandable, and it's kind of working. So as someone who is kind of in the middle between the two worlds of immune repertoire sequencing. I would strongly advocate to keep stuff in flat file format. Commas, tabs, I don't care. If you look at, and the point was made that once you have to put many entries into a tab, right, it can get out of hand with commas, but really you don't care if you can just parse this, right? Like whether your tab is 15 meters long, right, as long as it's comma separated. So I would want to be a strong advocate for keeping stuff in a flat file format that your tool exports, and if you then want to build on this and re-import this and do stuff with it, that's fine, but at least keep that option. You know, you would lose me. I don't know whether that would be horrible, but you would most certainly lose me in the process.
I think that flat file is incredibly important and is not going to go away. I would not say that people in genomics are very happy with the state of things with parsing multiple file types and having to do conversions, and it sucks when your life is 80% converting data from one format to another. So I think tools like VDJML help avoid that because they take advantage of data standards. I mean, really, there's two things you're talking about. You're kind of really talking about what is the content of the format? What are the fields we want to support? Do people need to have an ability to even represent numbering or not? And then there's the totally independent question of, once you've decided what goes in it, do I store it as XML? Do I store it as TSV? Do I store it in some kind of binary format?
That's what I was going to say. So, and so like yesterday I said, I came up here and go, well, can't we just accept VDJML and move on? But I guess the real question is, can we accept the ontology behind VDJML? And then those are just targets, and then whatever format and tool you use, you just have to, those are the targets, I think.
One other comment, I mean, maybe if we settle on a particular file format, we could just require that any program that gets the blessing of AIRR has a conversion both to and from. And so you can convert anybody's data into whatever format you use, and you can always convert it back. So kind of like a universal language. And each of the programs could work with whatever file type they prefer.
Yes, I think this was brought up a little bit just before, but I think one of the keys is going to be, defining the column names for the flat file format or these element names, in the XML. I mean, the idea when we sort of developed this change of format was to allow multiple tools to work together, right? So the idea is you could have this data file, you could run a cloning method on it, and it would add a column that said, this is the clone ID. And then your tool for diversity calculation would know that there's a column that should be called clone ID, and it could use that data in order to calculate diversity. And that way you could string multiple tools together from multiple different groups, and they would know which columns to look for and which columns to output. So I think regardless of which way we go with VDJML, Changeo, and we're writing converters between the two, that's going to be the important thing that we as a community can come up with. What are we going to call these column names, these elements, and what's going to be the definition behind them? Yeah, agreed. And then just to pick up on what Brandon said, it is in our recommendation list that we want to have tools be interoperable, and just like Steve was saying about his own tools and adding the column, that one of our hard recommendations to go back to that is that people should be making their tools in a way that you can get output, whether it's direct output in a common file format or whether it's, as you said, offering a conversion. But that is definitely already in our list of recommendations. And I think that Steve's point is good for focusing the conversation a little bit, because the way I read the question that Eric wrote here is what other things do we need? And maybe we also, as Steve said, need to agree on what the column should be titled, so that anybody can read it in. But what columns do we want is also sort of the same thing.
Just a slight addition. I will note that when we were talking about VDJML, the schema design, we took a lot of information out of the IMG ontology. So that would be a good place to look for things that you think might currently be missing.
So there's a lot coming up. I think maybe I'll just move us on. And just to close, I mean, I do want to say that I think that what the work that the group behind VDGML has been fantastic, and I think it's really important to define these ontologies. So let's see. So I sat down with Corey last night, and we sort of tried to decide what are the sort of base things that if we could magically modify IMGT, what would be the sort of core things that would be really needed? So the first is certainly clarify the licensing issues. So we need to be able to build on top of this resource. So there's been various suggestions about how we could legally make a workaround using GenBake IDs and say that these happen to coincide with things that are present in the IMGT database. I don't really want to go too far down that road, but that has been suggested. But sort of the two really other core things are being able to include novel alleles derived from cDNA sequences and developing reasonable criteria for including novel alleles inferred from repertoire sequencing data. And I guess I should also add, it would be wonderful if we could have some explicit versioning plus the ability to sort of roll back to previous versions. So do people have any comments on this?
Should we not have minimum standards for what we think a database should look like in the first place, rather than simply try to improve an existing database?
That's the next slide.
So we had kind of consciously divvied this up into things that we would ask for with maybe the possibility that they would be accepted as changes, and then on the next slide we have other things that we might do independently of IMGT. I don't know, should we just move to that? Unless Steve's got something to say. Excellent.
I guess just to throw something concrete out there, I know many of us have been maintaining our own local versions of novel allele databases. Maybe as a first step, I would propose that we make a central place, maybe based on the database that Andrew was describing yesterday, where we all agree to deposit our novel alleles.
So the idea would be this list is sort of designed to avoid having to do a new database. So if IMGT agrees to accept these novel alleles that people have been accumulating, that sort of strikes me at least as ideal in some sense. And then if not, then yes, we definitely need to do it to have a different place to store them. And either way, though, then we're going to need annotations on those sequences, metadata, whether they're on IMGT or stored with Andrew or somewhere else. We're going to need a metadata layer on those that is the next slide. So here's what we've come up with from discussions in the working group about types of useful metadata. So obviously the type of evidence, whether it's from gDNA or cDNA, from Sanger or NGS, whether it's a direct observation or an inferred germline, how many times was a number of observations here is number of donors, I should say, and how many different independent studies. We might, we would want to add as meta annotation the CDR and framework boundaries to the extent that we can agree on them. That's actually the one piece of metadata that really is in IMGT currently, but if we're adding sequences or storing sequences somewhere else, we would also want to have chromosomal positioning, synonymous names, because if we have to start our own database, then we're going to have to still be able to cross-match with the IMGT names, or even just as a way things currently exist, if IMGT renames 1-F to 169.2, we want to be able to keep track of that. Some sort of versioning, this could be either at the database level or as a metadata of individual sequences. Something that we came up in the sort of extensive debate last night is that we probably, even if we're accepting inferred sequences for some level of inference, there may still be some things that we want to tag as provisional versus some, and then on the flip side of that to be able to deprecate. So even if we create a place where we say we're comfortable with having inferred alleles as part of the database, there may still be cases where the inference is weak, or it's only, or one of the cases we talked about last night, if it's very different from any known sequence, it might be, is it receptor editing or something, so we might want to be able to tag things provisional, and likewise deprecate if need be. And so this is the list we came up with last night. We'd love to add to it if there are things that people think need to be added.
So there was a question raised about minimal standards for such a database, and I think this is exactly the right time to be talking about that. In particular, you've got type of evidence as the first item, but I would go further than that and say not just type of evidence, but the strength of the evidence as well. And of course you said that, but I think that it's really important to get that out there, and perhaps to have a working group that works on exactly what, how to weigh evidence and to come up with a way of marking things with particular level of confidence.
Right. So this was, I think that's true. This was specifically labeled type of evidence because I was making very basic distinctions in terms of, what data source is it coming from or so on. Strength of evidence, I think we would have to think a little more carefully about. So strength of evidence would also include the next category, the number of donors that's been observed and the number of independent studies that have seen it. And it might include nuances of the type of evidence, not just that it's Sanger versus NGS, but how is the NGS prepared and the error curve. So I think that you're right, but this was meant to be a first step, a more basic step, where just a categorical approach, and then we may want to do something that's less discrete and more nuanced as a layer on top of that. Absolutely.
So Steve and I both walked up at the same time, and in fact I was partly going to direct a question to him in that one measure of the strength of the evidence is for an inferred sequence is the rearrangement frequency that you observe, and obviously the rarer the utilization of a particular sequence, the tougher it is to call. And I'm not sure what kind of limitations are presently seen with TIGA, but it might be, and of course I'm also interested in variability in the utilization frequency, and a really neat database would actually record observed utilization frequencies, which would both illustrate the diversity of expression, but also would give you very concrete evidence of the strength of the evidence.
I guess I would say sort of the strength versus type question is very important because I think one of the limitations is right now we can say a lot about the different types of evidence that we have, sort of where it comes from, how many times we see it, how many different individuals. But I don't actually think we have enough information and data to assess strength of evidence. I mean, we don't have enough cases, I think, where we've inferred things computationally from these kinds of data sets and then really went back and validated them that these alleles really exist and they're really there. And we probably need more data and more research to really sort of translate between sort of type of evidence and strength.
So as a first approximation, you could do the ratio, the magnitude of the ratio in TIGR, for instance, could be, like you said, it would have to be validated. Does that really correlate with anything? But it might be at a first step, it might be considered.
Right. So we have tons of stuff that intuitively makes a lot of sense that it should give you stronger evidence that this is a real allele, but it's just not been fully validated in most cases.
Yes.
Just to add to that a little bit, though, I'm not disagreeing necessarily, but layer on that, I think we have seen both alleles that TIGR found and that the Collins group found have been genomically sequenced now. I would say a large proportion of them, right? I don't know if that helps, but
it helps, but it's not quite enough yet, I don't think. And I think that the question really for us is, do we want to endorse a germline database that at this stage essentially allows anything to be put into it as long as you say whether it was inferred or directly observed? Or is it important to wait until we have developed the appropriate validation criteria? I mean, so my concern is that once the cat is out of the bag, it's going to be very difficult to rein in and say, oh, by the way, we no longer believe that these particular alleles are valid.
So that's an important question because I think as a working group, we've been basically assuming the former scenario, and or as probably is obvious that we've been sort of assuming that the former is preferable. But yeah, if people have strong opinions on that, then that's a very important thing to point out, Tom. And part of that may also be in terms of the cat getting out of the bag is we have said, oh, we should have tags for provisional and a mechanism for deprecation. But actually what those are, especially the mechanism for deprecation, is going to be important if we're going to do it this way, where we're going to say, right now everything goes as long as you annotate it properly. So that still strikes me as a better approach, but yeah, if anybody thinks that we should take the other option and try to be more measured here, speak up, please.
I think the one thing that we should add to this is that, of course, if we decide to set up a new database, what will be very important to have a governance structure that is really based on the community, because otherwise this will be just a change of masters, and in 5 years we will be complaining about the next database curator. So therefore, I think what we probably could use as an example are, for example, the CD standardization committees that immunology has used. So basically, people can describe stuff, submit it on a provisional basis, and then there are regular meetings which then weigh the level of evidence, whether things are in or not, which then would go well along with the structure that Chaim already explained, so that we specifically consider that everything that is in this database in theory can also be deprecated at some point, because we simply don't consider it valid anymore, or we think that it's a sequencing error and it was just a copy of another gene. Just to note that I mean, this is also a question for tool providers, that if we provide this metadata, does this give you enough information to incorporate the germline in your tools? And I guess also, would you be prepared to do that? Would you be prepared to update the tool on a regular basis, or to allow people to specify additional alleles if this information is provided?
I guess we would, and I think this is, very important. And I think commonly what a lot of us do, we'll take sort of the reference, germline allele set from someplace like IMGT, and combine it with a local set of alleles that we have, that we found locally, and that we sort of collect from people like Scott Boyd, who are willing to sort of send their novel allele sets, and sort of concatenate all those together as something to reference against. And I think, again, one way to handle it is to start putting something like this together as a place where we can collect together these novel alleles. I mean, we certainly believe very strongly in the alleles that we infer through Tigger and other methods, but maybe sort of have that as a staging area where they can be, further investigated. Other people look for them, find those same alleles in lots of other individuals, and then as you get more and more evidence, maybe then it goes into try and push that into IMGT or some other sort of really strong reference set.
Yeah, so this goes back to the question that Tom asked, and I think that one of the goals here is for reproducibility. So if I say, I did an analysis of this with germline set X, that somebody else can then find germline set X. And one of the reasons I think it might be better to go with a catch-all approach is precisely that if everything always goes into some central location, and then we can put versioning dates on it, deprecation flags, and so on, and then you can say, I used the germline database revision 102, or with SH5, whatever. That might be better. We have talked on the BTCR forum a little bit more about how this might work mechanistically, whether sequences would all have to be in one place, like on a GitHub or something, or whether actual sequences might live sort of distributed and just have a sort of along the lines of how the common repository for raw data has been talked about, and that is, I think, open, but although maybe less of a priority than determining what we want in the database in the first place. But that's I think the important point is if you do an analysis with a germline reference set X, I want to be able to go back and get that somehow. And so that's one of the reasons for putting everything in a big bucket to start with, and worrying about sorting it out with metadata and version numbers later.
I just wanted to, Christian's going to go next, but I just want to suggest that there's a real opportunity for automation here. I mean, if we are talking about pulling together a lot of data sets as part of the AIRR effort, why not have a bot that is continually scanning these new things for alleles and and that's why I asked about objective criteria yesterday. I mean, we at the end of the day, I hope that we can formulate our criteria into something that one could reasonably type into a computer program, and then that's, for me, the true objectivity.
That's a great idea. I really think that is something that we should be moving towards, and I think that the discussion about how we would validate the inferences that are being made now would be the first step towards what you're describing, Eric. And I think that's a fabulous idea. We should be able to just scan all the data that comes up, just as you're saying, and do the discovery automatically. Brilliant.
Okay, so just because since we're reaching the end of the session, I mean, in the end, the letter that was drafted, or these points that were drafted by Eric and Corey, I think there should be one thing that should be pointed out. This, in our terms, is not some kind of feeble feature request to IMGT. This is the last and final call. And therefore, I think it's very important to see whether we, as a community right now, would really support this if this has been spelled out in a nice and appropriate way and would be a public letter to IMGT. So therefore, my question would be, as far as these points go, would you see yourself be able to sign such a letter? Okay, so everyone who's not, would you not sign because you think that this is not appropriate due to IMGT's past performance, or do you think that IMGT is past reform anyhow and that this wouldn't change anything? I mean, we don't have to bring this up, but I think this is, it's important to see that we try to speak with one voice before we make that decision. Okay, thanks. I guess we will bring this up tomorrow again.
The session does go to 12:15, so I wanted to just point out a couple last things. So maybe this is just my own personal thing, but I hope that in the future of AIRR session we can discuss how we can engage the public in what I really think is a fascinating type of research. There's a thread about this on BTCR, but basically the microbiome community has been amazing at pulling together funding and so on, coming from a lot of outpouring of public interest in this subtopic. And personally, I think that everything that you can say about the microbiome, you can say about the repertoire, but more so. So I hope we can have some brainstorming now and next year and in the future about how we can try to generate this type of excitement.
The best thing to get the public's attention is to have stories about patients, particularly little children that you've cured,
I'm completely serious. That's how you do it. And you know, the thing that really captured the public's interest about the microbiome, for instance, was that it was the microbiome that was responsible for the fact that you're fat, not the fact that you know, eat chocolate cake all the time. For example, that obesity study was one of the very first microbiome things that came out, and that was just huge, and it went everywhere. So it's got to be stories about people, not stories about databases.
Well, I mean, yeah, you people, yeah, or people who are naturally immune to certain things, or people who respond to vaccines and people who don't.
They also have an amazing citizen science type outreach thing where you can send your poop in and get it sequenced and stuff like that. And I mean, I really would love it if some company would do this. Except the problem is, that's the problem.
Just to echo what Danny said, I mean, the fecal transplant Carl Zimmer New York Times story also was but that was a huge microbiome thing, right? And now there are DIY fecal transplant websites, which is terrifying. But no, but I think these types of broad reach out science writing engagement with the science writing community and stories about people and the talk we heard yesterday morning about, you know, Dr. Rosenberg's talk was amazing. Those are the cases that we need to highlight in a more broad-based approach. Definitely.
Yeah, I was just going to say, if we can just have Steve Rosenberg talk about repertoires enough, then we'll be on the map.
I think one of the big distinctions with the microbiome is that everyone feels like they can now take some probiotic, right? It's like we want to find out why I'm fat because of microbiome so that I can easily change it. But I think like this kind of fundamental like difference in the story is that it's harder to find ways to I just
go get your flu shot.
I'm a selfish American, so how does this affect me?
Go get your flu shot and you'll see how
So we need to vaccinate people against obesity, is what you're saying.
Damn straight.
This is the point where we're going to degenerate. We just need Donald Trump to tweet about us.
So speaking of the next session, we have 4 talks and 1 hour. So, I mean, I love your idea. I don't know if we're going to get there, but we could try. So I just want to ask the working group leaders, can we all meet just right before lunch at the end of this to talk about just exactly what we are going to discuss in the next?
No.
Yes, we can. Not you. You can't come.
Oh, okay, good.
Yeah, I mean, sure, okay, we can break, but I mean, like this was the thing that sort of started me. Okay, this is all the elements that we need, right? Michael Pollan, like, he's like this. Oh, he wrote this book, Omnivore's Dilemma. He's a food guy, and like, you know, here's Michael Pollan, and here's all his gut microbes and how they compare with other people. Like, I mean, yes, I agree that we need stories, but I also think that we need to be able to have maps like this.
We do have a couple of new visualization tools. We might need to encourage somebody who's more creative than me to develop other cool visualization tools, but there are a few out there.
Jamie, please use the microphone.
Jamie, there are 20. There are 20.
There's a UCSD guy. He's who's that guy, Felix? You come to the microphone. No. There's a guy who's done all this stuff on...
Are you talking about Rob Knight?
He's like a computational guy. We went to his meeting. What's that guy's name? At UCSD. Smart. UCSD.
He's not older anymore.
No, he did everything on himself, and then he presented it for 2
Smar.
hours to us.
David Smar.
David Smar.
Yeah.
He's our guy.
That's a good point. Okay, well...
Careful what you wish for. Like, honestly, because the meteor is a double-edged sword, right? So my last paper, I'll brag here, it was commentary in Nature that was with a few other people, and our story that was on, you know, fairly dry regulatory affairs span the world with homebrew heroin as the headline, right? So I became the queen of homebrew heroin with some colleagues at MIT. So regardless of whether you're talking about repositories like really boring dry stuff or studies in mice, it's a journalistic frame to always start with a human interest story. So you're going to end up with a human interest story whether you want it or not. And you just, working with the media, it has all kinds of repercussions. So in the stem cell world, there's all kinds of hype that leads to really poor behaviors around stem cell clinics and other things. So I would just, yes, engaging the public is important, and yes, media is important, but please do it responsibly. Just my little...
Great. I mean, let's have a round of applause for all of you.